next speaker and closing speaker, which will be Dr. Vincent Anidiobu from the Food Process Engineering section in the Department of Food Technology at the Federal Polytechnic University of Ado-Ekiti in Nigeria. Vincent's work will be around the solution to honey adulteration in Nigeria, and he will present more in detail a rheological model, which I believe might be also translatable to other states. A little bit about Vincent. Vincent was born 42 years ago in Enugu, southeast of Nigeria. He obtained a bachelor's degree in engineering or chemical engineering in 2003, went on to do a master's degree in sciences in 2008, and his PhD in 2017 from the University of Lagos. He is a member of professional bodies, including the Nigerian Society of Chemical Engineers, the Nigerian Society of Engineers, the Nigerian Institute of Food Science and Technology, and the German Rheological Society. He is also a registered and chartered engineer with the Council for the Regulation of Engineering in Nigeria, and his passion and expertise is in the field of rheology. Vincent serves as a senior lecturer with vast experience in food engineering and food technology department at Ado-Ekiti University. And he is also within the university, the chairman of the Committee on Open Distance and Flexible Learning. He has certifications on open educational resources, flexible skills development, Moodle learning, and by the Commonwealth of Learning. He is happily married and has children, and today we are going to dip a little bit deeper into rheology.
Thank you very much for the compliments. Yeah. We're looking at the solution of honey adulteration in Nigeria, a rheological method. Thank you so much for the compliment. By way of presentation, we have the following presentation outline, which we deal with introduction, theoretical background, experimental results and discussions, conclusions and recommendations. By way of introduction, we know that honey is a sweet fluid produced by honey from the nectar of flower. In this work, we'll be looking at honey as a sugar solution so that we'll be able to follow the rheological properties that we follow suit. Honey is formed when bees gather nectar, thousands of worker bees gather nectar, and then regurgitate it in their honey stomach, and then deposit in the nectar, and it ripens over a period of time. The uses of honey transcend medicinal, nutritional, even industrial. In this study, we are looking at rheology. Of course, we know it's the study of the formation and flow of fluid. Rheology, in this rheology, it will aid us to get an intrinsic property of honey. Intrinsic in the sense that it will lead us into the composition, molecular weight, and even molecular weight distribution of honey. Of course, we are looking at this study in the light of whether it's a simple fluid in which the viscosity is dependent on shear rate and time. Of course, it is not. And that is the definition of a structured fluid. For the statement of progress, we know that honey can only be produced by bees. In Africa here, we have bees, mellifera bees, which is very hostile. And there is high demand for honey. The high demand for honey makes it expensive. And the expensive nature of honey leads to illicit adulteration of honey to meet up with demands. In 2010, the world honey demand was about 1.5 million tons, while the supply is about 2 million tons, according to Food and Agricultural Organization. And the difference is always augmented by adulteration. The conventional method of honey adulteration detection is expensive and time-consuming, and that is why we are suggesting rheology. The aim of this study is to develop a test method based on rheology that can differentiate pure and adulterated honey. And the objectives are to explore the efficacy of utilizing honey rheology to assess its purity. Secondly, to extract structural, compositional information from the rheological data. Thirdly, is to validate the result of rheological characterization using established test methods. And fourth, to establish rheological characterization and testing as a testing method for honey quality. The scope and the limitations of this study is these samples were gathered between February 2010 and November 2012. For the significance of the study, we already know the high demand for honey and its low supply for the product. It promotes adulteration. And the existing method for honey quality assessments, like you can see earlier, they are expensive and also very time-consuming. The rheological method for the quality assessment of fluid will be desirable, being economical and simple. This study establishes the efficacy of rheological assessment for honey. The testing method deriving from this study will find application in the quality assessment of the other fluid. For the theoretical background, we have the development of structural kinetic model. We have the structured state in equation 1 when the honey is at rest. And then we have the non-structured state when the honey undergoes deformation, that is shearing. It will be assumed that change in the rheological behavior of honey is associated with shear-induced breakdown of internal structure of pure honey. This model we assume in accordance with the above equation that the structure of honey breaks down irreversibly under shear, but will build up completely after shearing. We have theta being defined in equation 3 depending on shear rate and time. Equation 3 shows here that the experimental viscosity of honey minus infinite shear viscosity, which is the viscosity at infinite time, all over zero shear viscosity, which is viscosity at 0 time minus infinite shear viscosity. So we have the decay equation. And in this decay equation will be solved. It's emanated from equation 1, just like we do it in the reaction kinetics. K here is the rate of deformation, and N here is the order of the deformation. N will tell us the number of active components that are actually taking part in the deformation of honey. So the structural kinetic model was solved as an initial value product, and we have equation 5. Equation 6 is Mark-Hawkins relation. This Mark-Hawkins relation is used to replace the zero shear viscosity in equation 5, and we have equation 7. Equation 7 here is designated as the structural kinetic model for the Nigerian honey. This model has some parameters that will be able to differentiate pure from adulterated honey. Chief among which is the molecular weight, which we are going to gain insight into using the flow properties of honey. Also in equation 8 here, we have the IAgrE model. IAgrE model has similar properties like the developed structural kinetic model. But in IAgrE model, we have the behavior index N. This behavior index gives us an introspection of the quality of honey. If it is less than 1, that honey is shear-taining. If it is 1, the honey is a Newtonian fluid, and it is above 1, it is a shear-thickening fluid. And also equation 6, which is the Mark-Hawkins relation, is used to replace the zero shear viscosity in the IAgrE model. So in equation 9, we have the amended IAgrE model. So this amended IAgrE model will be used to calculate or predict the molecular weight of honey from the deformational parameters. Also, we have in equation 10 here the power law model. It's a 2-parameter power law model. N here is also the behavior index, and N of O is the zero shear viscosity. The power law model was solved using a least square analysis, and we will see the results subsequently. In Nigeria, there is incessant adulteration of honey, and because of that, we couldn't really trust any honey sample to start with this work. we had to cultivate, harvest our own honey at Federal Polytechnic, Ado-Ekiti in Nigeria. You can see us by 8 p.m. on the 29th of November harvesting this honey sample. Why 8 p.m.? Why in the night? Because the Apis mellifera bees in Nigeria, in Africa, is very, very hostile. So we take nights to harvest here when they will be less active. So our sample H here is our control sample. Then we collected other samples, B1 to B7 from northern Nigeria. All of them were gotten from northern Nigeria. We got C1 to C6 from southern Nigeria. We also have to adulterate ourselves using high fructose corn syrup so that we can compare the rheological pattern of this adulterated honey with the ones we picked at random from all over Nigeria, so that we will be able to place them into what people are doing around. This is the sample demography of where we picked our samples in Nigeria. For the rheological characterization, we made use of group field programmable rheometer, having a cone and plate. Of course, the measurements were carried out using the cone radius of 24 millimeters and with a 0.8 cone angle. This will give us a gap of 0.1 millimeters and the circumference of the at the circumference of the cone. We worked at a very, very shear rate, a low shear rate in this work, where the molecular interactions actually drives rheology. So the samples were made to stand in the container for at least 30 minutes before the analysis. This is to allow for complete structural build-up from the shear in this flow into the container before the for the correlation test, we determined the hydroxymethyl content in honey. This content material will always not be present in fresh, well-prepared honey. And according to the Codex Alimentarius, in the tropical Africa, the standard is 80 parts per million. That's, but I understand that for the European Union, it is actually 40 parts per million. The difference there is because we have predominantly high temperatures here, of course, which leads to the building up of this hydroxymethyl. It is actually produced by thermal dehydration of exocysts, that is the glucose and fructose, producing hydroxymethyl as one of the byproducts. We also looked at the antibacterial properties of the Nigerian honey. In Nigeria here, honey is used in our hospital for the treatment of bed sores and on many antibacterial resistant microbes. So we decided to look at antibacterial activities of honey, since it's an intrinsic part of Nigerian honey. And we studied it using two organisms, a gram-positive organism, Staphylococcus aureus, and a gram-negative organism, Pseudomonas aeruginosa, obtained from University Teaching Hospital, Ado-Ekiti, using agar diffusion method, CSLI 2012. This is the rheogram of a pure honey. In this rheogram, you will see that pure honey at the inception of flow will exhibit a shear thickening behavior. There is a peak and there is predominantly shear thickening behavior. Like we said, we are looking at honey as a sugar solution. Triantafilopoulos, 1988, in trying to explain the rheology of some oligomers, he said that when the fluid is at rest, the particles of the fluid fits into the void of adjacent layers of fluid. But during the flow, as flow commences, instead of filling the voids, they slide over the void, leading to the observed shear thickening behavior. At the peak, the rate of particle association is in equilibrium with the rate of dissociation. As flow commences, there is isothermal and reversible breaking of the structures of the fluid, which leads to predominantly shear thickening behavior observed for the Nigerian honey. This is the result of the effect of high fructose corn syrup on the viscosity of honey. We will see a sequential decrease of the viscosity of the observed honey. The green line here is 10% adulterated with high fructose corn syrup. This is 50%, this is 70%, this is 90%, and this is the flow of high fructose corn syrup. Then for the honey samples picked at random from all over Nigeria, north and south of Nigeria, you will see this sample B1 behaving rheologically like the structure, like this sample of our index case. Also that of B7, you can also see it here. The two are similar to that of, but the rheological models will tell us more. So this is the power law fit of Nigerian. You will see a lot of discrepancies at the inception of flow. So the power law model actually was not doing enough for us here. And that is why we transferred to amended Carrier-Yasuda model that gave us a very good fit. This is the 10% adulteration, that is figure 7. Figure 8 here is the 50% adulteration. In trying to use the structural kinetic model, we have a viscosity time plot from where the 0 time viscosity was extrapolated. From this zero time viscosity, the molecular weight was gotten. Also, we have we use the coefficient of variation to actually extract the infinite time viscosity, because we worked at a very low shear rate. And with the time we worked, it wouldn't have deformed for a very long time for us to get this parameter. So we have the result of the power law model. We see here for the pure honey, the behavior index was 0.25, showing us that it is actually a shear thinning fluid. And this behavior index increases with increasing adulteration of honey. This is the result obtained for the Carrier-Yasuda model. You will see, most importantly, the molecular weight of honey obtained here to be 205. One would have expected that the molecular weight of 180 would have been obtained from honey, because it's a mixture of mainly glucose and the fructose. But this honey contains melizitose and also raffinose, which are oligomers. And these oligomers are the ones that are driving the rheology of honey. So their presence in honey actually impacted on the molecular weight of the obtained pure honey. Then we have for the fructose here, one would have thought that the molecular weight would have been 180, going by that of ordinary glucose, ordinary fructose. Of course, glucose is an oligomer of it, of an isomer of it, sorry. But the 159 was obtained, showing the presence of water content impacted on the result. We also see a sequential decrease of the molecular weight of the obtained of the adulterated samples with their increasing adulteration. Also, this is the result obtained for the structural kinetic model. You see the molecular weight of pure honey being 202, similar to what we got using structural kinetic model. Actually, the difference in their similarities was because of the minor error, the error differences between the curve fitting in using those two methods. Actually, we saw here that the order of breakdown kinetics here is reading trade for honey. That's what we obtained for honey. It suggests here that there are 3 components in honey driving its rheology. The number 1 there is water. Number 2 there are the monosaccharides. Number 3 there are the oligomers in honey, which I've previously mentioned. And you see that the order of kinetics actually reduces with increasing adulteration. You see that when it was only fructose alone, you see the order to the 1.5 is actually reflecting that majority of what is taking part in the deformation is what is fructose with some element of water going on there. So this is a classification of the result of the samples we picked from north and south of Nigeria using our method. you will see that only 3 samples out of the whole lot were classified to be pure here. And that is B1, B7, C4. All others had some elements of adulteration. Most importantly to look at is sample B4. Sample B4 is not completely honey. We later discovered that it was just fructose impregnated with honey flavor, and it's being sold in Nigeria as honey. The implication of this is that some people who bought honey for medicinal purposes will have their sicknesses prolonged. So this is the result of a correlation test for the hydroxyl methyl furfural. You will see at the retention time of 4.073, we have that is for the pure hydroxyl methyl furfural, and that is compared with that of honey with the same retention time, comparing their peak areas. And the result for pure honey was obtained there to be 1.43, showing that this honey sample is actually fresh and also not adulterated. These are the results obtained, which correlates closely with the rheological characterization of honey. As we can see, the pure honey is giving an infinitesimal quantity of hydroxyl methyl furfural, and so on. So this is the result of antibacterial effect on honey using Staphylococcus aureus. We saw a clear zone of inhibition from the Nigerian honey, but we discovered that this zone of inhibition decreases with increasing adulteration of honey. And after 70% of adulteration, the sample will no longer be able to perform any antibacterial properties. A similar result was obtained using Pseudomonas aeruginosa. So these are the summary of the findings. From the first objective, it can be found, or it has been found, that the viscosity of pure honey decreases as shear rate increases. The rheology of honey from Nigeria reflects it in its composition. We are saying also that rheology is an excellent tool for honey characterization, since the flow behavior correlates closely with the compositional changes in honey. For the second objective, we have here that the behavior indices in the power and the law model and Kariushida law models, we are the distinguishing factors as they increase with the level of adulteration. Also, a structural kinetic model was derived here, and the model was used to predict the average molecular weight of honey as a means of further distinguishing pure from adulterated honey. The new model was able to predict the changes in composition of honey serially diluted with adulterants. The order of deformation in the model tells the number of active components taking part during the deformation. The best result for pure honey was obtained, as I said previously, at third order deformation kinetics. We also have here that the Kariushida model was improved upon as a tool for the determination of molecular weight of honey, just as a secondary assessment. And this compared relatively well with that from our new structural kinetic model. For the third objective, the result of the antibacterial and chromatographic characterization of honey correlated well with the new rheological method. For the fourth, a new testing method for honey for its authenticity and quality has been established. And this method was able to detect adulteration and form a calibration for serial diluted honey in accordance to their levels of adulteration. Other honey samples picked at random from all over the country were classified to their levels of purity using the outcome of serial dilution. In conclusion, the objectives of this study, of course, were achieved. We are concluding that at room temperatures, 27 degrees Celsius in Nigeria, pure honey exhibits shear thinning flow pattern at low shear rate. Why did we use low shear rate? At this low shear rate, the molecular orientation, the molecular interaction actually govern the observed results. But as share rate increases, just like you can see in previous results, honey will give you predominantly a Newtonian behavior. So compositional changes in honey due to the addition of common adulterant reflect in this sample's rheogram, suggesting rheological characterization as a method for detecting adulteration of honey. By extension, rheological characterization can serve to detect faking of the product. Power law and Career-Sudan models can measure the degree of adulteration in honey. The structural kinetic model developed in this study produced plausible estimates for the average molecular weight of honey studied, and by this suggests itself as a sample quality tracker. The result from the amended Career-Sudan model compared relatively well. Of course, we've said that before. For contributions to knowledge. These contributions to knowledge actually have been published in some journals recently this year, Anidiobu 2021, published at Atello and Francis Journal of Apiculture. We also see Anidiobu 2016, Anidiobu 2014, and all the like. Recommendation. This is very, very important. In view of the limited number of source distribution of samples used in this study, the conclusion drawn can be augmented to a more general applicability by including and analyzing more samples from locations outside Nigeria. The result of these samples should also inspire the design and manufacture of a simple test apparatus for the rheological screening of samples for authenticity and quality. Thank you for listening. Before I stop, I would like to acknowledge three people. The first among them, they are from University of Lagos, Dr. John Wallo and Dr. Faith Bambola. They were my teachers. They were instrumental to guide me to grow in the field of rheology. Also, I want to acknowledge here the rector of my institution, Engineer Dr. E.H. Oladebe, for giving me the latitude to be part of what you're doing. Thank you very much.
Thank you, Vincent. I got a couple of questions in. The first one is non-rheology related, so maybe one of the colleagues from today earlier will jump in. Kelechi is asking, interesting to know about the hostile nature of the African bees. Though not related to rheology, are there less hostile species in Nigeria?
Actually, in Nigeria here, we have Apis mellifera. And I want to pinpoint part of the adulteration we have in Nigeria to be as a result of the bees. A lot of people here in Nigeria want to go into honey production, but the bees here are very hostile. So they're very difficult to manage. So that is what we have for now. But in future, the European species of bee can be brought here and make our life easy in honey production. Thank you.
Yeah. A practical question. How easy is it to adapt your rheological model for other countries? And the second question that came with it, do you think there are plans to use the rheological method for national standards? In Nigeria, I assume.
Yes, for the rheological method, we've actually suggested that to immediately after my PhD work in 2016, we suggested that to National Agency for Food and Drug Administration and Control in Nigeria. Actually, you can see, and I want to acknowledge them to some of the slides I showed them. They ran those samples for me free of charge. We have recommended that to them, but we are yet to get feedback from them. Maybe we may have to write a reminder to them and also write also standard organization of Nigeria so that this rheological method can be used as a standard for honey quality assessment in Nigeria. Also, the person asking the adaptability of my method in other countries, I would like to see that. I would like to see that. I would like to be invited to come and demonstrate that. Thank you very much.
Okay, Ayodele is asking, well, first saying good presentation, compliments. So the question is, how was the honey collected from the different regions and kept before the experiment? Could this have affected the viscosity of the honey itself?
The honey were actually collected from the beekeepers and kept in the refrigerator at 4 degrees Celsius in Nigeria. And we got it transported like that to our labs. And it was retained at that temperature shortly before our analysis. Of course, we don't really keep them for a long time. Immediately we get those samples, we analyze them, we get the results. So that because currently we are even looking at the aging, the effect of aging on the rheology of some samples. Thank you.
Okay, another question. Are you aware if there are any do-it-yourself DIY test kits for measuring honey rheology available for Nigeria?
No, I'm not. No. I do not. We are still working. We are still working. And that is actually the focus of this work. If you look at my recommendation, we recommend just a simple test apparatus that will lead to, that differentiation. We are working on it. Thank you.
Yeah, I got another question from Jay. Have you cross-referenced the results with other methods, like the ones we have seen earlier from Sandra, for example? Or did you purely use GC?
Sorry?
Like, have you cross-referenced the results of your rheological testing with other methods?
Methods like?
I guess like some other chemical analysis, like I guess GC or HPLC or.
Of course. Like the determination of hydroxymethylfurfural it was done using the method of Julian and Cooper's experimental techniques, 1980. It's an HPLC method. Actually, we are working on GCMS to look at the sugar assay of Nigerian honey. We've also looked at the use of protein content, water content, but for this study, I just picked the most potent parameters for this study. Thank you.
Okay, Longinus is asking, please, did you use the 100% imitator adulterated honey as a negative control?
Of course, we have our sample B for there, which was a negative control. From the onset, from the smell and sensory evaluations and everything, we knew that it was a negative control. So for that negative control, it gave us completely Newtonian behavior. Yes, we did.
And the last question I have, I think it's more philosophical one to everyone to think about from Anthony. As a worldwide question, who is to blame for the world supply of honey as far as its adulteration? That's a good question.
Yes, we blame ourselves. We blame ourselves. We are supposed to work more. We are supposed to work more. We are supposed to produce more honey, especially in Africa, where adulteration is thriving. So we blame ourselves, and also the environment created by the government in this part of the world is not conducive. So that is the major blame. So if we have enough grants and enough encouragement, of course, we can flood the Nigerian market with good honey. But currently, we don't. Thank you.
A question from Folasade. Thanking you for your presentation and asking, can a layman, anyone basically identify adulterated honey easily? Or does it really require these rheometrical analysis and chemical analysis?
Okay. For a layman, this is not 100% correct, but it will go a very long way. If you get a good honey, turn it over, allow it to flow, allow the flow to become thinner and thinner and thinner and become the thinnest. If it is good honey, it's not likely to break into droplets. Actually, I'm trying to think like the machines. It's not going to break, but this is not actually very correct, because you start getting this at about 70% adulteration. So you can see, it's not there. So for now, there's nothing much we can do for the layman.
Yeah. Okay. I mean, I want to thank everyone for today. I forgot to mention we actually accompanied today by our CEO, Charlie Nicklin from the Institution of Agricultural Engineers, who kept in the background. If anyone has interest in membership within the institution, we have a website, IAgrE.org. If anyone wants to follow up with any of the speakers today, you can just drop me an email. I will just broker the contact, get you all together. I dropped my email also in the chat function a little bit earlier. And last but not least, I think, I don't know if you all want to unmute our microphones and just give a little bit applause to our speakers.
Yeah.
Well done. Well done.
Yeah.
Well done, the speakers. Good job.
Yeah. Charlie, do you want to say a word? No.
Unmute myself. Yeah, I mean, I apologize. I haven't seen all the presentations because I had a prior commitment, so I've only really caught the back end of it, but it looked really interesting. And as Daniel said, if anybody's interested in joining our institution, please have a look at the website. But yeah, nothing more from me, Daniel. Yeah,
perfect. Oh, actually, I see Anthony was from the US. Oh, well, that must have been torture for him today. Yeah. Thank you, everyone. Yeah, as said, a few things. Everyone who signed up through the Eventbrite link, I will produce some attendance certificates, which you can use for your CPD log if you do so. Just give me a few days. Given the numbers, we had 177 registrations. And as said, if you want to follow up with any of the speakers, I'd happily link you up. Just drop me an email or anyone else of the IAgrE team, and we take it.
I assume, Daniel, I assume Sarah said it was recorded as well. So if people wanted to look back at any of the footage, they can do.
Yeah, she said. I think it's actually you who needs to stop the recording now.
Yeah, I will stop the recording now. Okay.
Okay. Thank you.
Thank you.